Hello everyone, and welcome to this mini lecture on enhancing absorption with lipid-based delivery systems. Today I will discuss emulsions, colloids, liposomes, and phytosomes as delivery systems that effectively enhance absorption of natural products and pharmaceutical agents. Today we have four major learning objectives. First, we'll define two major factors that explain poor absorption. Then we'll talk about when to consider a lipid-based delivery system. We'll also review key facts, pros and cons of the different delivery systems. And of course, we'll talk about how to pick the best option. Don't get excited about that because you might be a little bit disappointed by the punchline. I highly recommend you check out the webinar A Medicine's Journey, A Visual Introduction to Pharmacokinetics. Knowing that absorption is just one component of bioavailability, and bioavailability is so important in understanding how to choose the best therapy for a patient. To summarize the key points from this webinar, we talked about a medicine needing to get to the site of action in the body. And to achieve that, it has to get in, it has to find the target, it has to stay intact, and then it has to get out. These four parameters determine the efficacy of a medicine from a quantitative and temporal standpoint. You might see ADME in the medical literature, you might see it in textbooks, you might see it in papers. Now you know what that acronym means. It's worth mentioning that good absorption doesn't always matter. It depends on the ingredient, depends on the compound. So I encourage you to take a look at this really quick article that goes through different scenarios when good absorption actually doesn't matter. Absorption is defined as the process of crossing the gut wall. It contributes to bioavailability, which is defined as the fraction that reaches the bloodstream, or more rigorously, the fraction that reaches the site of action in the body, which tends to be hard to measure. So typically with bioavailability, we settle on the former definition. The other ADME factors determine how much of the compound reaches the blood and the target tissue and how long it stays there. So we are talking about the first step in a very long journey. You may remember from biology class that a cell membrane serves to protect the interior of a cell from its outside environment. Membranes are lipid bilayers that are made out of phospholipids. And phospholipids are ideally suited for this job because they have a polar head group that's hydrophilic or water-loving, and they have a fatty acid component that is hydrophobic or fat-loving. So naturally, when you put phospholipids into solution, they like to form a structure and organize themselves in a way that orients the polar head group towards the water and the fatty acid tails towards each other. Phospholipids are amphipathic, meaning they have both a hydrophobic and a hydrophilic part. In the Medicine's Journey lecture, I talked about four ways to cross a membrane. There's passive diffusion, which doesn't require any kind of carrier. There's carrier-mediated transport, which requires a protein that carries a compound across a membrane. There's efflux, which takes the compound and spits it back out of the cell. And then there's paracellular transport, which slips between cells. The majority of phytochemicals and many drugs rely on passive diffusion. Many vitamins and minerals, on the other hand, tend to require carriers. The exceptions would be the fat-soluble vitamins, which can cross a membrane through passive diffusion. So today we're only going to be focusing on passive diffusion. This process has several advantages. You're simply slipping right through. It's a non-saturable process. No carrier is needed. No genetic variations are going to affect this process because there's no protein involved. But you need to have a small particle size, and you have to have the right solubility. And you also need a sufficient concentration. You have to have a lot of compound reaching the membrane surface because this passive process is driven by a concentration gradient. So a high concentration on one side of the membrane is going to allow the process to go in the correct direction. You're going to drive a lot of that compound across and get into the cell. So here I've illustrated the process of absorption. We're going to assume that this is the small intestine where most drugs and dietary ingredients are absorbed. And the membranes are located in the intestinal lining or the epithelium, which is a monolayer of mucosal cells. And to cross these membranes, a compound has to meet certain physical and chemical criteria. If it's too hydrophilic, it's probably going to hang out on the outside of the membrane and not get through. If it's too lipophilic, it might not be able to penetrate this water layer and even access the mucosal surface in a high enough concentration to capitalize on a concentration gradient that's required for passive diffusion. And therefore, it's no surprise that the most effective drugs and dietary supplements tend to have a moderately lipophilic character and a small particle size, which we'll get to in a moment. So the key point to remember here is that a bioactive has to be soluble enough in the aqueous environment of the GI tract in order to be absorbed. This is a problem for fat-soluble vitamins, vitamins A, D, E, and K, phytochemicals like terpenes, triterpenoids, curcuminoids, flavonoids, and many other natural and synthetic therapeutic agents. So the key take-home point here is poor absorption is often due to poor solubility. So we all know that oil and water don't mix. So how do we get these lipophilic compounds into an aqueous solution? We can do this through emulsification or creating a colloid. So these are systems where you're mixing immiscible liquids or liquids and solids and creating a uniform, homogeneous mixture. So these are commonly applied in pharmaceuticals and nutraceuticals as a way of solving the solubility problem, and they are very effective in doing so. So nature has its own way of solving the solubility problem for fat-soluble vitamins and other nutrients. The liver produces bile, which is stored in the gallbladder and released into the small intestine. And bile salts are amphipathic, so they have a lipid-soluble and a water-soluble component. And that allows them to break up large lipid droplets into smaller ones. And that makes the droplets easier for digestive enzymes, or lipases, to access and digest. So this process not only improves solubility, but it reduces particle size. So pharmaceutical and nutraceutical emulsions recapitulate this kind of process and provide a solubilized bioactive that wouldn't normally be water-soluble. And often phospholipids are used in the manufacturing because of their amphipathic character. So that's why you'll see lecithin or phosphatidylcholine or something similar on the label for a lot of those products. Emulsions are defined as a mixture of two immiscible liquids, like oil and water, whereas a colloid may involve solid particles that are mixed in. So subtle difference, not important to know. Just remember that both systems are appropriate ways to improve the solubility characteristics and absorption of many different types of bioactives. Emulsification, whether it's occurring in vivo or whether it's occurring in a manufacturing process, creates small droplets called micelles. And micelles are these spherical particles that enclose the oily core or the fat-soluble compound, and the micelle surface is water-soluble, so it easily crosses the water layer in the intestine and accesses the mucosal surface for absorption. Here's an example of a colloidal system where we have coenzyme Q10 in the Vesisorb delivery system, which is a self-assembling colloid, and the Cmax of CoQ10 was significantly higher for the Vesisorb system compared to these other formulations. Now, take note that there are two solubilized coenzyme Q10 formulations that were included in this study, and the Vesisorb delivery was superior to both. There was a twofold increase in peak plasma concentration between one of the solubilized coenzyme Q10 formulations and the Vesisorb system. So not all colloidal systems and emulsions are created equal. There's a lot of diversity out there as far as the efficacy of the system. So always request pharmacokinetic data whenever you're considering one of these advanced delivery systems. I will mention that the Vesisorb system has been successfully validated for a variety of lipophilic compounds, including cannabidiol or CBD. Here we have results from a randomized double-blind crossover trial in which a single oral dose of 25 milligrams of CBD was given to healthy volunteers, compared to a standard formulation where CBD was just mixed with medium-chain triglycerides. CBD in the colloidal system had a much higher peak plasma concentration, a much higher area under the curve, and a faster absorption profile. So clearly there was an advantage of the colloidal system, but this is a very specific patented technology, so don't assume that because it just says colloidal on the label that this kind of data would apply to the product. If you're not familiar with the three terms I just used, I encourage you to check out this short video where I define Cmax, Tmax, and area under the curve. This will allow you to interpret pharmacokinetic data with more confidence. So when do these systems make sense in clinical settings? If you have a patient with bile insufficiency or fat malabsorption, you might want to take a look at what's available as far as emulsions and colloids go. Whenever there's poor compliance with a meal for whatever reason, if you want more flexibility throughout the day for dosing, you generally don't need to take emulsions and colloids with food because you're not depending on bile for emulsification. Whenever there's a liquid delivery preference, you might be able to find an emulsion. Some of these are available as liquids. And topical therapies typically require some kind of an emulsion or colloid. This is outside the scope of today's talk. So I've listed a number of examples of compatible bioactives in which these systems have been successfully applied. Keep in mind that this is just a partial list. I wanted to briefly mention the pros and cons of these systems. The pros would be widespread use, enhanced solubility, reduced particle size, so we're satisfying those two criteria that were our primary goals for these enhanced delivery systems. And then the cons would be stability issues with emulsions. You always want to check the expiration date. They tend to have a shorter shelf life. You can definitely expect variations in efficacy depending on the company that you're sourcing your product from. So now that we've covered emulsions, let's look at liposomes. And liposomes are much more sophisticated. They are microscopic vesicles in which an aqueous interior is enclosed by a lipid bilayer. You don't have just one layer of phospholipids. This is a bilayer, and it's a lot like a cell membrane. There's no chemical bonding. The phosphatidylcholine simply surrounds this water-soluble bioactive on the inside, and hundreds or even thousands of phosphatidylcholine molecules may surround the drug. So you have a lot of phospholipid in the formulation. You'll see phospholipids or lecithin or something similar on the label for these products. Unfortunately, you won't be able to tell whether it's a true liposome by looking at the product label. So you really have to look into your manufacturer and make sure that there's been structural validation of the liposomal structure. Liposomes can be used to deliver water-soluble compounds, which would be on the inside of the particle. We can also use them to deliver fat-soluble compounds, which can be loaded into the membrane itself. So they are much more versatile. You can also engineer liposomes to have a very small particle size, down to a nano-sized particle. So that could be highly advantageous when you're trying to maximize absorption. Here's a quick example of an absorption enhancement with a liposomal system. This is an in vitro model of the intestinal lining. And here, glutathione was more effectively absorbed across the cell monolayer compared to a glutathione control, which was just a standard powder. So peptides like glutathione are great candidates for liposomes. Here's an example of vitamin C in a liposomal format, outperforming a non-liposomal and placebo in a human study. It should be noted that in parallel, they examined intravenous vitamin C, whose bioavailability was off this chart. So the key takeaway here is never rely on just one graph as your sole point of decision making. You should at least read the abstract and familiarize yourself with the design of the study and learn what the y-axis means. Are these units significant in terms of the incremental difference? So these are things that you really want to pay attention to when you're trying to compare different oral formulations. So when do liposomes make sense? Anytime you are administering a peptide like glutathione, it does make sense. Glutathione is inherently problematic because it's subject to degradation by gamma-glutamyl-transferases and other enzymes in the small intestine. Malabsorption, any kind of bile insufficiency is also a good reason to consider a liposome. And anytime you have a non-responder, somebody who's tried glutathione or something before and has not responded, you might want to consider one of these advanced delivery systems. Examples of compatible bioactives are quite extensive, but in general, whenever you're looking at a peptide to administer orally, liposomes make sense. Vitamins, phytochemicals are also compatible with this system. Here are the major pros and cons. Lots of pros: high solubility, enhanced permeation. You can get down to a very small particle size. Some liposomes can protect the bioactive from degradation by digestive enzymes or even metabolic enzymes. And of course, decades of research in the pharmaceutical industry has given us a robust proof of concept. There are three cons that you have to keep in mind with liposomes. First, they're going to be more expensive. They're also going to differ depending on what company you're obtaining your liposome from. No two formulations should ever be assumed to be equivalent. And also, you want to get structural validation. Structural quality assurance may come in the way of electron microscopy or some other imaging technique that proves the presence of true liposomes. Let's move on to phytosomes. You'll notice that the structure of a phytosome looks very similar to that of a liposome. And like a liposome, we have a bilayer of phospholipids, but the main difference here is that the bioactive is enclosed in the head group of the phospholipid, not inside the particle and not inside the lipid bilayer. This allows a greater stoichiometric molar ratio of phytochemical, for example, to phospholipids. So theoretically, you could potentially cram more bioactive into this particle compared to some liposomes. In terms of functionalities, there are a lot of similarities between phytosomes and liposomes. Enhanced absorption across membranes is made possible by the optimized solubility characteristics of these particles. There might be some resistance to degradation by enzymes, so you might protect the bioactive. And phytosomes have been extensively validated for a variety of phytochemicals and herbal ingredients. Plasma curcumin levels were about 20-fold greater with a curcumin phytosome compared to unformulated curcumin. There's also been quite a bit of human data published looking at specific clinical endpoints, for example, joint function and CRP levels. So to reduce CRP levels and improve joint mobility, for example, you typically need gram doses of curcumin. But in a 3-month clinical trial, a very small dose of 200 milligrams of curcuminoids per day in a phytosome format reduced CRP and improved joint mobility. So 200 milligrams compared to a gram dose is a huge difference. Here's a pharmacokinetic profile of quercetin in a phytosome. Whether given at a dose of 250 milligrams or 500 milligrams, the phytosome clearly outperformed the unformulated quercetin by a landslide. So you can see a clear advantage with area under the curve, as well as peak plasma concentrations. Reasons to consider phytosomes would really be the same as for liposomes and emulsions. Whenever you have poor absorption, for whatever reason, bile insufficiency, fat malabsorption, whenever you're dealing with a non-responder who tried quercetin, for example, before and didn't respond, you would want to consider one of these advanced technologies. A phytosome would be a great match for quercetin. Compatible bioactives for phytosomes are probably more extensive than what I've listed here, but they include quercetin, milk thistle, ginkgo, OPCs from grapeseed extract, green tea, hawthorn, and ginseng. With phytosomes, the pros definitely outweigh the cons. Good solubility, good permeation, good digestive stability, small particle size, excellent shelf stability, more predictable would be an important pro. Unlike liposomes, phytosomes are generally made by one manufacturer. So if it says phytosome on the label, you know exactly what you're getting. This is the clinically validated technology. The only two cons with phytosomes would be price point. You can expect to pay more for a phytosome, and the fact that this technology has only been applied for a select number of botanicals and phytochemicals. So now you understand the key functionalities and differences between these different delivery systems, and you might be wondering which one is best. I hate to disappoint you, but it's up to you to evaluate the pharmacokinetic data on a case-by-case basis and avoid making assumptions based on the delivery vehicle alone. The best format for a given patient case depends on a variety of factors, and it always boils down to what compound you're dealing with and what limitations it has in the absorption process. I'll conclude with 5 take-home points. First, poor absorption often results from poor solubility and/or large particle size, and these problems can be effectively addressed using lipid-based delivery systems. Emulsions improve solubility, but the pharmacokinetic advantage is less predictable because no two emulsions or colloids are the same, unless you're dealing with a patented technology like Phytosome, for example. Liposomes, and particularly phytosomes, are more strategically engineered and are likely to be a little bit more predictable. Always beware of liposomes that lack structural quality assurance data, such as electron microscopy. And finally, always evaluate the data for each individual product to make a good decision as to whether one of these lipid-based delivery systems makes sense for a given patient. I highly recommend this additional learning resource. This will prepare you to interpret bioavailability data and make the best decisions in clinical practice, and you can test your knowledge with the study questions. I hope you found this information helpful. Please feel free to submit feedback, questions, or comments through my website, integrativepharmacology.com, and be sure to subscribe. Thank you, and see you next time.